So we have already noted that we have different mechanisms through which you have cells getting what they want and getting rid of what they don't want. So that means that we are looking at, we have looked so far at the processes through which cells get what they want and remove what they don't want. So that is an issue to do with movement in and out. And we have already noted that actually there are two broad categories of these mechanisms, where that is, these can be active transport and also passive transport mechanisms. And in this case, we are going to look at these processes basically in detail so that we understand how they happen in cells. So we'll look at this question during a lab simulation. So a simulation is when you have someone demonstrating what could be happening in a particular physiological process. Okay, so during a lab simulation of the immune response, students observe white blood cells engulfing bacteria. Okay, but through what process these white blood cells engulfing this bacteria? How do they do it? Okay, and what happens to the bacteria when it has been engulfed by the white blood cell? So here we're basically talking about how the white blood cells basically capture this bacteria, destroy it. What processes are those, and things like that. So we are basically introducing concepts to do with endocytosis and how it happens. How is it different from exocytosis? Do these processes actually require energy? And if they don't require energy, why? If they require energy, why again? And things like that. Can we say that endocytosis happens in nutrient absorption in cellular organisms? If so, how does that happen? Those are questions that we should be able to answer. So we use a broad term called cytosis to describe the ways in which the organisms form what we term as vesicles. Okay, so do they capture a substrate and turn it into a vesicle? What process is that? Okay, is what they have captured, is it in liquid form? Is it a solid particle that they have captured? What structures do they use for capturing? So cytosis is a form of active transport involving vesicle formation, and we shall look at that. So when you have let us say waste products in a particular cell, you will have vesicles being formed, and then that vesicle is the one that fuses with the cell membrane, and then the contents are released out. Okay, so you can have endocytosis when you have something being captured from out the cell and then becoming part of the cell, or exo, a vesicle that was inside the cell and then it opens into the surrounding and releases the products out. That is exocytosis. But of course, we need to look at them in detail. So endocytosis. So you have a cell creating a depression, what we are terming as an inward folding. So this is the cell. And then creates a kind of mouth here, and then it captures. It can be another pathogen, it can be a food particle, captures it. That is what we call endocytosis, taking it in into that cell. So it can be by phagocytosis, pinocytosis, or receptor-mediated endocytosis, when we have a specific molecule that is being captured because it has fitted well into a particular receptor. So let's again look at this. This is what we are basically explaining. You have a particular receptor that captures particular molecules that can fit well onto that receptor. And when they fit well, you will have inward folding being initiated, and then eventually you have a vesicle that is formed, and that vesicle will now enter into the cytoplasm. So you have a cell with its vesicle. Okay, and then phagocytosis is what we can term as cell eating, that for example, the white blood cell looks for where the pathogen is and creates an invagination and then captures it. It engulfs it, okay, through the pseudopodia. You have the pseudopodia forming and then capturing it. That is what we call phagocytosis. So we have cells that we say they are phagocytic. So some of the white blood cells are actually phagocytic cells. That means that they use phagocytosis in dealing with pathogens. Okay, so what happens is that they form pseudopodia around the pathogen and close them up, and then become part of this cell, and then this is broken down. Okay, and if they are nutrients, they can be absorbed into that cell. Okay, so the plasma membrane forms vesicles, what we can term as phagosomes. Phagosomes fuse with the lysozymes. These lysozymes are the ones, lysosomes, okay? The lysosomes are the ones that will basically break down the contents of this material that has been, and then the waste, good things are absorbed, the waste are removed out. So the white blood cells basically do that. They use structures that we call the pseudopodia, those extensions that basically capture the microbes. And they take them in, break them down, remove what they want, get rid of the waste. Cytosis, basically the process is the same, but here we are dealing with smaller vesicles, and we are talking about liquids. Okay, so that's why we are saying cytosis is cell drinking. Sometimes you can have receptors being involved for uptake of particular kind of liquid substances. Exocytosis, exo meaning out, so getting rid of things into the surrounding environment from the cell. So remember, talking about vesicles or vacuoles. These vacuoles are formed within the cell. That is, say that the waste materials of the cell collect into the vacuole, and then this vacuole has to fuse with the cell membrane, and then the things are released into the surrounding environment outside. Okay, and then the vesicle membrane becomes part of the plasma membrane, and this is what secretory cells basically have. So exocytosis, the process is basically very simple. We are saying that you have to have vesicles forming, and these vesicles can contain a number of things that need to be removed. Now, these vesicles are forming from the Golgi apparatus. So these vesicles now move along what we term as the microtubules to the cell membrane, where you have that fusion. And then when that fusion happens, you have the content being released, and then that vesicle membrane becomes part of the plasma membrane. So this bulk movement of this content is very important in capturing pathogens, in capturing nutrients, and also in getting liquid components through pinocytosis. So white blood cells use phagocytosis, which is a form of cytosis. And then you'll have, when you have the vesicle membrane fusing with the plasma membrane, it means that the plasma membrane is being replenished Okay, and then formation of the cell wall also through this process, especially exocytosis. And then also the release of the acetylcholine, which is a neurotransmitter, is also through cytosis, basically exocytosis. And then the same thing happens with secretion of hormones. So many important things are released by cells through exocytosis. So we're talking about hormones, we're talking about neurotransmitters, and then we're talking about